Okay, and let me share my screen. Share screen. Okay, let me go to slideshow. Okay, can you see my screen, and can you see it in full screen and the pointer?
Yes.
Okay, perfect. Okay, thank you everyone for joining. Today I'm going to talk about this really nice paper from a collaborative group led by Dr. Connard from Harvard. And the talk, I picked this paper because it directly relates to two of our projects. One of them, the review of epigenetic rejuvenation, the epigenetic reprogramming for rejuvenations, mechanisms, and pathways. And the other one, it relates to our epigenetic analysis of epigenetic rejuvenation. So this is a work on epigenetic reprogramming, focusing on intermediate steps, early stages. And this group has been working on this, on understanding reprogramming, mechanisms of reprogramming for the last 10 years. Since 2012, I'm going to refer to those work and mostly focusing on how it relates to our work and how we can leverage this work. So if you, just to give you a quick overview, this is how we started thinking about the reprogramming in the context of rejuvenation. So our goal is to understand what's happening early in reprogramming that results in the rejuvenation phenotype that we see. So we know there are some Yamanaka factors being the key, and some enhancers that we use in our protocol. And they have some direct or indirect targets. And we've seen MIR302, AP1, they are being differentially expressed, and they have some transcriptomic or epigenetic effects. And through these effects, they are inducing some early or late cell processes. And we think somatic repression and chromatin remodeling and change of cell cycles and proliferation are earlier based on the literature and our own understanding. Metabolic shift, MET transition, we don't know where they are, and pluripotency comes later. And through these cell processes, they result in some rejuvenation effect that we reported in the paper, and some pluripotency effect that we want to avoid. So this was the original roadmap that we came up with. And since then, we've been reading the literature and doing analysis, producing data to understand this process better. Okay. So last time, this is the structure of our review paper. We have 3 sections: reprogramming factors that I presented last time. It's also recorded and available online if you want to watch. Then the second section of our work is on reprogramming early events, and Son is leading that part. Here I'm just talking about a few pieces of that section and how this paper relates to that. And that's the other section. The third section is reprogramming for rejuvenation, and this is focused on the experimental studies that show rejuvenating effect. And now we have around 20 studies in vivo, in vitro, in human, in mouse. And Andy talked about this a bit last time, but probably we'll have another talk on this when we get closer to publication. And today, it relates to the second part, early events. Just to summarize what we did last time we focused on the specific factors. We went deep on each one of these factors and what are the mechanisms of action. So what are their structures? What are their binding, and how their structures help or explain their mechanisms of actions and their functions. And for each one of them, multiple potential pathways and mechanisms have been proposed, and we still don't exactly know which one of them is early, which one of them late. But one way to summarize them is together they induce pluripotency, they maintain pluripotency, mainly through autoregulatory networks. And we know this is late, so we want to avoid this for our rejuvenation study. We know they induce lots of chromatin remodeling from very early all the way to very late. So this is maybe the main effect that we want to study better. And we know there is also these effects are coupled with proliferation from early, because mainly we have MYC. And it's recently shown that MYC could be removed. But what we did last time, we talked about these individual factors or how they work together, for example, OCT and SOX, and how they have these actions, direct actions that could relate to reprogramming. But the last question Vittorio asked last time was, okay, now how they induce these reprogramming events. And this talk is talking a little bit, getting a little bit into that, how they relate to these events that we are interested. And this is a nice figure prepared by Andy, putting everything together. This is one of the figures of the review paper. And if you have any feedback on this, it could help us to make it better for the review, or any questions so far. I'll take a pause here to see if there's any questions about what we did last time. So the idea is to have different domains of these factors and to have their DNA bindings or motifs, and then to have a 3D structure of these factors all in one figure for the paper. Okay, great.
Madi, sorry, is there, probably not, but has anybody ever done like the crystal structure, for example, of OCT and SOX together, or is there anything like that, or probably not? It's too difficult.
I think out of all these, we have one crystal structure of OCT and SOX. Andy, do you remember anything about that?
Yeah. So on the right, basically that's it. So on the right is I took the picture from the PDB, which is the protein data bank. And on the right, you can see the crystal structure of in particular, what they did was the binding domain of OCT4, the binding domain of SOX, and the nucleosome. So on the right, if you see the SOX2, there is like OCT4, POU, and there is SOX2, HMG.
Oh, sorry, I didn't see that.
Yeah, this one is basically, these are the two binding domain that are highlighted schematically in the center, and how these two together bind to the nucleosome. And if you look at HMG binding domain, it's basically the same of POUH. It's just reverse complement, because see, it's ACA, AA, TG, and on the other side is CT, TT, GT. So basically what was found is that the two, they bind the same sequence, and also the binding motifs of OCT2 and OCT4 are kind of overlapping. So based on crystal structure, it seems that these two together are able to bind to the nucleosome, forming a 90-degree angle. That's what basically open the nucleosome. That's why they're considering as pioneer transcription factors in the process.
I see. So so if I'm reading this correctly, so you have the single so the single crystal structure, I mean DNA protein complex for OCT4, KLF4, and CMYC, but you have both OCT4 and SOX2.
Correct.
For okay.
Yeah, OCT4 is a homodimer, so because it's chain 1 and chain 2. These are the one available that were discovered. So probably when they incubate together OCT4 and the DNA, apparently OCT4 dimerized, form a dimer, and bind the DNA.
I was sorry. The reason why I'm asking is that I was wondering if for consistency, it would make sense to see SOX2 alone.
There is no. Yes,
There is not.
point. Good point. It's not available. No. Yes, that's a good. Yeah, that's what I was looking for. But the only crystal structure available is the binding between SOX2, OX4, and the nucleosome. It's the only structure available.
And
binding domain, not the whole protein, because you can see only the binding domain of the DNA in the crystal structure for all of them.
And there is nothing about NANOG and Lin28. Well, Lin28 is not a transcription factor, but there is nothing about NANOG.
I didn't find anything interesting, but I didn't check a lot. It might be, but since in the section we are describing the main OKFM, and we are just describing briefly the other cofactors, I didn't put them. But if you want, I can check.
I'm just thinking because you know, the two pioneer works on iPSCs were done with OSKM and OSKLN. So those were the two first human IPS papers, and we also use 6 factors. So for our cocktail. So I was wondering if it would also make sense to add NANOG and also add the Lin28 and show that it's obviously regulating the microRNA machinery.
So out of all the final studies that you found, all the experiments, most of them, maybe 90% of them, use the original OSKM. And since we don't have much space, we decided to just focus on OSKM, and that's like now a consensus of the main factors. And most of the review papers also focus on OSKM, and because there are also lots of other enhancers besides Lin and NANOG. Now there are other strategies, even more efficient, proposed to be more efficient, and we thought it might be enough to, based on the space, to talk about these 4.
There was at the beginning a paragraph, other cofactors, but there were a lot. So because of the space, we said, okay. We described Lin and NANOG in the partial reprogramming section, Rejuvenation, just describing that these two seems to help and to make the process more efficient.
Okay.
Okay, great. So now for part 2, we have early reprogramming events. And the main events could be categorized as somatic silencing. And so if we look at the whole reprogramming event, the 2 big events is to silence in the reprogramming for pluripotency, silence the somatic factors for expression and turn on the pluripotency factors or a pluripotency network. But focusing on early reprogramming, one of the main events becomes a silencing somatic or somatic silencing and chromatin remodeling also as an event, and then metabolic shift and met transition possibly. But the main 2 that we are focusing on is somatic silencing and chromatin remodeling. So the main challenges in studying the reprogramming event and early reprogramming event is it's really finding a needle in haystack, because we have to decouple rejuvenating versus other events. There are many events happening all together. We are inducing these pioneering factors, these master regulators. They are doing all sorts of things. So first you have to decouple this rejuvenating event out of many events, sometimes overlapping. And the other one is decoupling rejuvenating cells from other cells. And typically we have a very few percentage of the cells that go through reprogramming and become IPS. And we think, personally, I think those are the ones that are becoming younger. It's for debate, but still, just to study them, it's a challenge because it's a minority of the cells. So now the proposed solutions are either to study poised iPSC, which are the cells enriched for poised iPSC, and study them. These are the cells that are going through the path of reprogramming. So we can separate these cells from other cells and compare them and study them separately. So that's the strategy. that's used in this paper, the paper that we are focusing on today. And another groups are working on making the rejuvenation or reprogramming less stochastic. And they are working on deterministic mechanisms of reprogramming and synchronized reprogramming. So the second part, the second strategy works on event. And there are other papers that are also giving us interesting insight based on that strategy. But today's paper focusing mostly on poised iPSC. And the way, this is the big picture of the paper. It's mouse embryonic fibroblast. And most of the cells, the bigger arrow is refractory. And they don't become iPSC and they are not going through the reprogramming path. But the smaller percentage of them, typically 1% out of all embryonic fibroblasts become iPS typically. But if we enrich for poised cells, we can improve the efficiency up to 50% by day 3. So day 3, these enriched cells, 50% of them become iPSC. And by day 6, 95% of these enriched cells become iPSC. So studying these cells is like studying the cells that are the small portion of the cells that are becoming iPSC and are going through the reprogramming path. So we can study these cells and compare and see what is different between them, what's happening to them. So that's the idea of the paper, the main advantage, the main solution to study reprogramming. But it's really hard problem. So this is 2018. So their first paper is 2012, and it's still even more recent paper. It's a very ongoing challenge.
Madi, sorry, it seems that tell me if I'm wrong, according to what you see and the literature, the poised one are the one that undergo rejuvenation and reprogramming, while the refractory one, they don't seem to be affected. If we measure the rejuvenation using, I don't know, whatever, epigenetic clocks or something.
Right, we don't have a detailed study on that, but my feeling and my overall understanding is that these cells that are going through reprogramming, they are also becoming younger like. So we don't know about the other path. There is no study on that. But if I have to bet, I would choose these cells that are becoming younger. But I think Vittorio still is open on this. Last time we talked. So
yeah,
Vittorio,
quite honestly, I think that no one has ever really looked into that question. I mean, we are using reprogramming here as a measure of success.
Right.
And yes, reprogramming and rejuvenation for sure happen, during the course of IPS kind of reprogramming. But the fundamental question is like, what happens? And it can still be true, because you remember we talked about this. It can still be true that what we are seeing is an accelerated reprogramming, and just by the fact that we stop it, we prevent the IPS reprogramming from happening. But that doesn't mean that the cells were not uniformly going through that process. Okay. So that's something that I'm very agnostic about. So it would be very nice to see, to find a way where, maybe as you continue reprogramming, you look at the cells that are not making it through, and see what happens to them. Because it's very different. One thing is looking at the cell through reprogramming, right? So you have two populations, one that successfully reprogram and one that doesn't. But we have always historically looked at this bottom population, right? The ones that make it successfully. But we have never looked from a rejuvenation standpoint, let's say, at what happens to the unsuccessful ones. So I think it would be very nice but then the question is, how do you look at that? I mean, if you stop the reprogramming, you are forcing the cells to not reprogram. So it's kind of an artificial, system, right? which was the foundation of our hypothesis. Like, but it would be very interesting to see as the reprogramming progresses, you know, to really focus on the ones that are not making iPSCs, because that's really the population that we want primarily to focus on. And even if you find that it's actually because of the successful reprogramming that the cells rejuvenate, which is possible, figuring out a way to prevent the reprogramming from happening. I mean, like the IPS reprogramming from happening, you know, which can be done by interruption, or you know, other ways. I don't know if it makes sense.
Yeah. I think it makes a lot of sense. Sorry, another layer to think about is kind of the stochastic nature of what becomes a successfully reprogrammed cell versus what becomes a refractory cell. And if you really look at the differences, and I have like no data to support this, obviously, but if you look at the differences, I think you'll find that cells that reprogram successfully to rejuvenate would probably have a more amenable chromatin landscape to begin with, versus the cells that are becoming refractory and sort of resisting the reprogramming. And I think it's going to come down to some regulatory regions that OSK and MYC are likely to bind in the early events, that the cells are sort of, you know, because if you look at the epigenetic landscape of cells in any given bulk sequencing data, you're really looking at aggregates of the signal, right? And you're not really looking at the individual cell and the chromatin sort of snapshot or the landscape in each given cell. And I think there would be actually some logic to the stochastic nature of what becomes a successfully reprogrammed versus unsuccessful or refractory. But I think that's something that can only really be figured out by the deterministic events, that solution of just looking through each of the factors, activity and together, and are there poised chromatin landscapes or state that makes certain cells more amenable than other cells.
Yeah, great point. And to add to that, so if I have to pick, I would pick this, but there are also studies that shows that these cells could be reprogrammed. These refractory cells also could be reprogrammed with other conditions, and if you extend the reprogramming or increase the reprogramming efficiency, they are not unreprogrammable. They just happened because of heterogeneity, that not going through that path. But I think all of them are first exposed and stressed, or they are all of them are changing. And the other thing is these cells are also reprogrammable. So it's just heterogeneity, and different condition might also induce these cells to rejuvenation or reprogramming. So I'm not saying that we can't, we have to ignore these 99% of the cells. Okay, great point. And so to talk about this paper, I'm going to talk about a few of the earlier work. So this paper is 2018. And if we look at the earlier paper by this group, same senior author, and 2012, I think this was also done at Harvard. This is one of the pioneering work. It had a big influence on the field, but some of the aspects of this are heavily debated and being updated. So this is one of the first papers to try to lay out the roadmap of reprogramming. And the idea was to do enrichment to find intermediate states or kind of poised cells early in reprogramming, do RNA, transcriptomic analysis, histone marks, and DNA analysis and DNA methylation analysis, and put them together and see what's happening. So the bottom line of this paper was that there are two main points of activity, two bursts of at least transcriptomic activity, one early, and then in the middle, not much happening. And then at the end, another burst of activities, transcriptomic activity. And this is also a longer reprogramming protocol. It's earlier, So it might have been changed a bit with more efficient reprogramming, but still, it was nice to separate the earliest stage from latest stage based on transcriptomic. This paper suggested that DNA methylation happens only late. So they disregarded, following a study, mostly disregard DNA methylation analysis. But they also found that the bivalent domains based on histone marks are changing from early. So they put the focus on chromatin remodeling as an early event during reprogramming. And the way, so this is the base for today's study. The way they enriched for these poised cells was to identify these two markers. I call them T1 and S1. And T1 is a fibroblast specific marker, and S1 is a poised or intermediate cell marker. And if you look at day 0, all the fibroblasts, they are mostly positive, almost 90% positive for T1. And by day 6, some of them are becoming T negative. And by day 9, some of them, more of them are becoming S positive, and these are the intermediate state. And if you look at day 12, followed by 4 additional days of reprogramming, they are mostly S positive. And when they are OCT4 positive, they stay S positive. So this paper mainly suggests T as a negative marker of going through reprogramming, and S as a positive marker. And they showed that S is going up all the way, and then maybe slightly reduced, and then T is also becoming irrelevant. So 90%, and again, it was a low efficiency, but they identified T as the marker of fibroblast specific, and S as an intermediate. We're going to discuss a bit more about the protocol for the paper, but this gives us the idea and starting to think about enrichment to finding intermediate cells.
Madi, this is the work is done in mouse, but I was wondering, did they mention in the paper if there is like evidence that this mechanism might be also conserved in human?
So most of these studies are still in mouse, even recent studies, because they want to, the idea is to control for heterogeneity, to induce the factors by transgene, and we can only do that in mouse. And for conservation, we know that overall, the process is conserved. They are the same factor. A lot of events are similar. So the idea is to use the mouse experiment and then see if it's consistent.
Because I remember that, for example, leaf had like an opposite effect in mouse versus human. I mean, Vittorio, I don't know if it's right. I remember from my stem cell classes that there was like some difference in the reprogramming, in the induction, I think, or in the maintenance of the pluripotent state in mouse and human. So, I mean, since it's a very powerful platform to address exactly what you were pointing before, maybe if we found that this marker are also conserved in human, we can probably use the same platform that they've used to divide this two cell population. But we have to be under the assumption that the system also works in human.
Yeah, so similar studies by Yamanaka group have been done for human. I don't remember if it's the same marker, but similar.
There are ways.
Yeah. And the follow-up study, so Dr. Polo is the first author of the previous study. Then he had his own lab, I think, in Australia, but still same group of people using same platforms. Now, this is more recent, 2017, and they do more assays. So previous work was just transcriptomic, histone marks, and DNA methylation. Here, they mainly focus on chromatin accessibility due to their finding about chromatin remodeling. So the main focus is on chromatin accessibility, chip-seq, attack-seq. And similar to previous work, they find a big change early and another change of events late, and not much happening in the middle. But this is another way to think about what's happening during the reprogramming. Somatic elements are going down, and there are some transient events here that's going up and down at the end again. And there are also some pluripotency regions that are becoming accessible. So in the y-axis, we have accessibility. So somatic accessibility goes down early. So we should be interested in all these three events: silencing of or closing of somatic regions, opening of early pluripotency regions, and opening of transient regions. And again, we don't know where the rejuvenation come from. Is a combination of all or not, or one of them. But this is a region of interest, and this is another way to look at the events that's happening. Again, focusing on poised cell. So again, we have enrichment, we have facts for the same factors. We are looking at T negative. We are discarding T positive here. We are looking at T negative and S positive all the way until day 9. And here we are, for example, using data from day 3. And we have day 3 and day 3T is the one that we don't want. These are the cells that are refractory. But D3 is the cells that have the early marker of reprogramming. And now using data from D3, attack-seq and chip-seq mostly, chip-seq of OCT and SOX, we can compare what's happening at regions that are open from the beginning in fibroblast and closed in fibroblast.
Mario, sorry if I interrupt, sorry. So I was just thinking something regarding to this refractory and successful. So when was done partial reprogramming, for example, from the J paper. So if you overexpress the factor for 5 days, I mean, the cocktail is different, but usually you expect to have 95% of cells that are refractory and 5% of cells that are undergoing. But when you do a bulk RNA-seq analysis, the transcriptomic seems to be, I mean, is a bulk, but if 90% of the cell were refractory to the reprogramming, I would even expect a difference in transcriptional output. I don't know if it makes sense.
Yeah, so the idea here is to enrich for the effect by just looking at the cells that are marker positive to reduce
for
that
rejuvenation.
effect.
For rejuvenation, if we work under the assumption, which I think is the most reliable hypothesis, that the cells that undergo reprogramming are the ones that are actually rejuvenating, how would you explain that when we overexpress the factors in fibroblasts, that we do a bulk RNA-seq, if 90% of the cells should be refractory, why we can see the transcriptomic readout of the rejuvenation, the epigenomic readout?
Yeah.
I don't know if it makes sense.
Yeah, exactly. So one potential answer to that is the refractory cells are refractory for reprogramming, but they might also, so that's what I'm saying by they are exposed and they are changed. So they are probably going to the same direction as the poised cells. They are just refractory to IPS, and maybe they are going to that direction to a less extent. And that's why when we extend the exposure or enhance the exposure, they also become pluripotent. So I think to a lower extent, the refractory cells are also becoming youthful. Yeah. But if you want to study them, so I would say maybe study the ones that are poised. Personally, I think it's more likely to see rejuvenating effects in the poised cells, or see it more clearly.
An alternative explanation could be that the platform that we are using, probably 90 to 95% of the cells are going through a poised state. Because the mRNA IPS reprogramming is very efficient. Okay. Well, it's not, of course, it's not 100% efficient.
Faster.
But you can tell by the behavior of the cells that most of the cells, change morphology, change, if you keep going, right? So it's possible that we're comparing pears and apples here. I mean, in this protocol, only 5% of the cells go through reprogramming. With the mRNA using 6 factors, probably most of them go through reprogramming. You know what I mean? So
Yeah.
the two hypotheses, I think, are still open. Bottom line is that we see a population-wide rejuvenation. But again, is that because the reprogramming platform is so efficient that they're all going through and then you stop it? And so, of course, then you stop it, they cannot go further, so they reverse. Or is it because the rejuvenation is happening? So you still have the two populations, 5% and 90%, but for the purpose of rejuvenation, all of them are affected. Well, maybe the poised ones are more affected, but even the refractory population is going through rejuvenation, or at least that early phase of reprogramming, but then it cannot go further. Does it make sense?
Yeah, exactly.
We are there's still both open as possibilities, in my opinion.
Yeah, and we haven't found any studies on that yet. There are still more studies on reprogramming, and because we still don't know exactly how
and
reprogramming.
Probably it depends also on the cell type, right? San, your I mean, not your work, but your group work shows that cells that are refractory by nature to reprogramming because they're post-mitotic, right? They rejuvenate, right? So yeah. I mean, how do you explain that? If you think that it's only the poise that actually rejuvenate? The neuron, I mean, at least I don't know if you got into that degree of detail, San, in your group, but to my understanding, at least, you are dealing only with post-mitotic cells, right?
Yeah.
So have you ever tried to make and you kept the transcription on for OSK on for what, up to 3 months?
Yeah, something like that. Yeah.
There's
another part.
Yeah.
There's another person who's done that study more closely. I'm not as familiar with the details of that study, unfortunately, but I can definitely ask. Yeah, but my understanding is that it's highly dependent on cell type and what cell type that you start with, whether it's a post-mitotic cell versus a cell that can proliferate. And that's one of the things that that we also find that you need to focus on for the early transient events of reprogramming, right? Is that proliferation seems to be sort of a necessary or somewhat necessary step in order for the rejuvenation to happen. But I don't know what that means for, again, like post-mitotic cells, like you're saying.
Yeah. We also have to say that the literature also split into different opinions, because there are papers that show that the dedifferentiation is essential, a partial dedifferentiation. So if the cells lose partially their identity and then redifferentiated, they are way younger. And other people, like all the reprogramming study in vivo, that basically show that probably even if you clean the chromatin without going so late, you can still have rejuvenation. I mean, the Belmonte paper that just came out, I mean, they did like 12 months of treatment, but very transient, only 2 days on and 5 days off, and they still observe a huge rejuvenation effect. And in that case, you are not pushing the cells to go in the partial reprogramming state, because it's only 2 days on and then 5 days off. So I think, as Mari said, there is really few molecular knowledge about this. That would be, I think, super interesting to dissect these two populations and to study them to have an answer. Yeah.
Well, another thing to consider too is that with the starting cells, if you're starting with older cells for rejuvenation purposes, you're going to see that a lot of the cells have actually lost some of their signature epigenetic landscape, right? That defines or that helps define the cell type. And so if you are already starting out with a population of cells that have heterogeneous landscape that have muddled identity, at least at the epigenetic level, then the efficiency is going to be varied pretty differently. And I think the heterogeneity, sort of erosion of the landscape that you see with age is also pretty tissue specific. So that's another thing to consider, that starting out, depending on what cell type that you start out with and how much erosion has occurred with age, you're going to see different efficiencies for reprogramming.
And rejuvenation.
That's a very good point. Yeah, you're right.
Yeah, great point. Definitely. So that's why they are all working with embryonic fibroblasts. But interestingly, interestingly, to add to that, it also amazes me how all these different cell types from different species are reprogrammed with the exact same factors. So I would say there is also a large commonality in the pathways and networks, and we might be able to leverage that commonality and find a universal cocktail for rejuvenation.
In this paper, they didn't look at the Thy1 positive population, right?
The Thy1 positive is used as they looked at it in the new paper. In the new paper also a bit. I'll show you the results in a bit, but this is just one slide from this earlier paper, this 2017 paper, but their finding was very consequential for the field and for us who are writing the review. Based on their early stage data, using ChIP-seq of OX4 as a representative of the factors, and ATAC-seq, comparing these two. So we have ATAC-seq and ChIP-seq, putting them together, comparing open regions in fibroblast and closed region in fibroblast. They showed that in the regions that are closed in fibroblast, these regions that are closed in fibroblast, they are becoming the target of OX4. So comparing the length of closed and open, most of the targets of OX4 is in day 3. Most of the targets are closed in fibroblasts. That was a debate up to this point, I would say, and they resolved this debate. And now we know that these Yamanaka factors within the first few days, at least for the cells that are going through the reprogramming, they bind to closed regions. And this helps us to explain the silencing mechanism. So based on this and follow-up and other studies, we now think that Yamanaka factors, they silence somatic program by displacing somatic transcription factors away from the open regions in somatic cells and to these potentially transient regions. Does it make sense?
It does, but my question, to go back again, because I am maybe I'm dumb, but I am interested about the refractory population. So if we see that even in the Thy1 positive, the Yamanaka factors must be there, right? So what are they doing? They're just hanging around, having beers in the cytoplasm, or what do they do? I mean, they must be binding somewhere, right? So, but why are they not successful in reprogramming? And if so, are they still successful in rejuvenation? So that's what I really care. That's the question.
Yeah. I think that's a more advanced question that they haven't reached that point. They are just trying to understand what's happening in the cells that are going through. But definitely your question would be more relevant to our rejuvenation studies.
I mean, this is like complete conjecture, but you could also argue that those sites or those cells that are refractory are, for whatever reason, have lost sort of epigenetic signals that mark enhancers or putative enhancers that are important for somatic or for pluripotency. And the transcription factors are actually having a hard time binding those sites, right? I mean, that's something that we don't know.
We don't know.
No. We have no idea. But, it's either, the question is either whether factors are binding, and then but they're not exerting the desired effect and not being efficient in the cells because of whatever epigenetic environment it is in, versus whether the factors are binding at all. And that, I think that's something that hasn't really been addressed.
Yeah.
Or they might be slower. They
Yeah.
might take more time because, I don't know. Because
They do
lazy.
end up, yeah, right.
Yeah.
Right.
So
Because I wouldn't expect the two fibroblasts to be so different epigenetically. I see your point, San, a lot if you compare neurons and mesenchyma stem cells, because the epigenetic landscape is super different, and regions are way less accessible since they are post-mitotic and super differentiated. But the same cell type, I found it's difficult for me to imagine that their two fibroblasts are so different, despite the transcriptomic is almost the same, that a huge overexpression of the factor is giving two different readouts. So I think another hypothesis that might be also reasonable to think is that they just take more time. And if this is true, they should be still be rejuvenating at this point, because you might just see something later. Okay.
So I think the reason why I kind of don't necessarily think that they could be, they would be, pretty close or uniform, is also kind of informed by my work in neural stem cells. So when I, worked on neural stem cells in grad school, a lot of the things that I was looking at were quiescent, so not cycling, quiescent neural stem cells in the adult brain versus activated neural stem cells. Those are basically equivalent cell types, right? They're just in different cycling states. But what you do see is that there's actually transient chromatin remodeling activity that happens between quiescent and activated cells. And when you look at the cell population and when you look at the cells individually, you do actually see a lot of enhancers that are partially blocked or partially open. So those are the cell-to-cell variability. I'm of the opinion that cell-to-cell variability and the accessibility of regulatory regions is actually a lot more heterogeneous than we think. We tend to think with the bulk data. Yeah.
So Madi, sorry, just to go back, they didn't look, so D3 here means SSCA1 positive. They didn't look at D3T.
As I did not find a detailed experiment, a detailed analysis like this for the negative cells. And this is still in the paper from a year before, from the more recent paper. I'm going to go a bit deeper to the recent paper. Yeah. So this is only one slide summary of that paper, but we have a bit more. So maybe we see something over there. But for the review for our work, so far we have a lot of question marks, and we are saying a lot that we don't know and the field doesn't know. But I think this kind of, I know it doesn't directly relate to rejuvenation, but even though I think it's good to focus on what we know. Also, so maybe here is a good point to say that the debate on somatic silencing has a consensus, and we think, at least for the cells that are going through reprogramming, the POID cells, the earliest stage POID cells, the mechanism is we can actually support this proposed mechanism compared to alternative mechanisms that are suggesting that the Yamanaka factors bind early to the open regions and messing up with the chromatin state by targeting the somatic TF in place. So I think here is an opportunity to take side and kind of summarize the literature and say this is a consensus. And rather than saying all the time that we don't know, or the field doesn't know and we don't know. So that's what I think. We can discuss this later on, but this is one of the points that I left a large comment on the paper and we can discuss further.
Yeah, I agree with you.
Great. Okay, now this is actually today's paper. Now we have a few slides on this, but most of it, I covered most of the foundation. And this is a more comprehensive study. They have 3/4 MEFs, two transgene mice. One of them is 4-factor OSKM, and the other one is OKS with mCherry. So they are using 2 transgene. They also have the knock-in of the OCT4 GFP, and they are studying day 3, day 6, and day 9 early reprogramming. What they do is they fax at different stage. At day 3, day 6, they are inducing with docs, fax at day 3, day 6, or day 9. And then after separating these cells, they continue docs for the rest of missing period, 9 more days or 6 more days or 3 more days until they reach the same total length of docs induction. And then they look at reprogramming efficiency. They adjust for plating efficiency. After fixing all these, then they look at this subpopulation and see if now they can use these poised cells and study what's happening for the cells, or to the cells that are going through reprogramming. Okay, so here is some of the staining results. So again, they start with T1 and S1 as the established marker. So since 2012 and more recently, more closer to this paper, the S1 has been very debated as a marker, early marker of IPS. And the reason it's debated is the result that you see here. So here is day 3, here is day 6, and here is day 9. So if you fax the cells on day 3, you look at T positive and T negative, and then you continue the docs for 9 additional days. You see that T negative in general have more iPSC. So that's okay. If you look at day 6, if you look at T positive and T negative, these two are T negative, one S negative and one S positive. Again, you see a little bit more on T negative group. And if you do the same at T9, again, you see more colonies for T negative. So it's okay. It's confirming that T is a fibroblast, is a refractory marker. But the surprising thing is the S positive and S negative at any day, day 3, day 6, or day 9 is almost the same. So they don't see any difference in enriching for S1. And this was reported previously, and that was the surprising effect. And some were debating that S1 might not be the early marker. But then they did a follow-up analysis and work, and they said most of this effect are confounding effect due to replating, because when you fax, you lose most of your cells, and that's actually, you see less and less cells that potentially, and you have a lot of confounding for replating and all that. So they find a way to adjust for that. And if you don't adjust, you see in day 3, we have 6% efficiency, up to 6% for T negative, but in day 6 we have lower, and in day 9 we have much lower. So this is confounded by replating and fax, doing fax at different time. So this different time biases the result, this different time of fax. But with a way that they found to adjust for the plating, now they have good results for day 3 and day 6 at least. So now all these results for day 3 and day 6 make sense. And the conclusion is S1 indeed is a marker of is an early marker of the cells that are going through reprogramming, as the difference between S positive and S negative is significant after adjustment. For day 9, it seems they cannot adjust it. And for the rest of the paper, they don't talk about day 9. So that was the main improvement and the main confirmation based on platform. So the rest of the paper, they look at day 3 versus day 6, and they call these cells as efficient and these cells as inefficient. So comparing efficient and inefficient cells. Any questions here? Oh, okay. So
you are not muted, but your microphone is not working. So you can talk here. Sasha, I have a question.
Can you tell me again
What do they exactly do to adjust?
Yeah, so the main problem here is that if you remember from the previous paper, they let me see if you have any figure on facts. So here, let me see maybe a better picture on
Probably
facts.
the number. It's probably the number of cells that you can plate.
Yeah, so when you fax, you have to stop the reprogramming process. You have to take them out of plate. You have to do a lot of processes to the cells before replating them. And since you are doing fax at different time, this faxing at different time itself biases the process. So they are not comparable again. And what they do is they adjust for plating efficiency. They control for the number of cells. It's just kind of an adjusting. I didn't go to the detail, but after the adjusting, now the efficiency is comparable between day 3 and day 6 and between different groups. That's the key. And it's not a groundbreaking technology. It's just a simple adjustment.
So it's just for playing.
Yeah, but the confounding effect was about replating. And yeah, I don't think it's even technologically a huge change in the platform. Okay, now what they do next is they look at all the proposed markers of early reprogramming. They call it transient markers. They find more somatic markers, they confirm more somatic markers, they confirm more IPS markers, but the key is they also find more early reprogramming, markers of early reprogramming. And then based on these new markers or the confirmed markers, they can now enrich by day 3 for the proficiency of 95%. And at day 3, the efficiency of 50%. So now after enriching, after faxing based on these additional markers, so they are arguing that a subset of S1 positive could be enriched, and that are the cells that are for sure or almost for sure becoming iPSC. And now they have 2 groups, day 3 and day 6, efficient and inefficient, and the efficient ones are the ones that 50% and 95% become iPSC. Now they compare these groups. So I'll tell you about their main findings. One of the 2 important findings from the paper, and probably that's all the time that we have. And I also didn't go to the detail of all their analysis, but to Vittorio's question, comparing efficient and inefficient, they also have data on the inefficient, on the refractory one. Now we can call this as a refractory to some extent, compared to at least the efficient group.
Sorry, I have a quick question. I was wondering if, and I don't know if I missed this, I was wondering if the model of somatic closing by displacement by OSK factors, do those regions have the binding motifs for OSK? Is that something that they've looked at?
Yeah, different papers look at this, and some of them found they have more markers of KLF. Different groups found different results. There is no consensus, but I remember one of them found more marker of KLF, and they think that KLF, at least in the transient group, has a role in bringing OS and K. But there is also another group that's called early iPSC, and these are the sites that these somatic TF bind and stay, and those are in combination with OSK, and some of those are actually binding site of OSK, and OSK stays, binds and stays for the rest of the reprogramming. But yeah, in this paper, I don't think they looked at that specific question. Okay, so here and now, one of the interesting findings is on chromatin remodeling, and they look at constant open regions in somatic cells and compare it with other groups. They look at somatic specific regions compared with other group. Ectopic region, this is equivalent to our transient regions, and this is the early IPSC that I was just referring to, and then ISP specific regions. This is based on ataxic data. This is based on accessibility. So it's interesting to see, the most interesting piece of this result is that fibroblast specific regions, which is 20% of all the accessible regions in fibroblasts, they shut down right away in day 3, inefficient and inefficient region. So that might relate to what Vittoria was asking, what happens to inefficient cells. Even inefficient cells, 100% of this subset of fibroblast specific open regions, they shut down right away immediately. So that was very surprising and interesting to me. So all the cells, regardless, these are also the regions that actually stay close all the way to iPSC. So that was a huge difference. And then ectopic regions, open and close. So, and there is no specific pattern, I would say. There is not a huge difference between efficient and inefficient. But early iPSC, when they start early, so this is what I was referring to, they open early and they keep and they increase this at least in the efficient. They're opening, the accessibility increases at least by day 6 in efficient. And in this protocol, day 6 is still early. And they stay open all the way to iPSC. And then they compared and say the accessibility is different between the efficient and inefficient based on their markers that they found. But yeah, I think this kind of platform led us to understand, at least in one subset of the cells or one stage of the reprogramming, what's happening, and very, very helpful. Any comments on that? Any feedback on that?
So this seems this really seems to support the idea that both at least in the early stages, both the inefficient and the efficient are going roughly through the same process, right?
In the earliest stage, so all these, all the top 4 rows are the earliest stages. So this is day 0. All the following 4, I would consider them as earliest stages, and overall, we can say they are similar. But comparing these 2, for example, there is a group of accessible regions that need to be open for cells to become iPSC. But these regions, exactly, these regions only open or mostly open in the efficient group. So that might be one of the key difference of these 2 groups.
Yeah, no, and again, you know, this makes a lot of sense in the context of successful reprogramming. The question that still remains open is, okay, that's great, because you know it's in a way it's a failure mode for reprogramming. But does that mean that the cells are younger?
Exactly.
Maybe they are. Maybe they are. Yeah.
I think what's more relevant to rejuvenation is this part, is 20% of the somatic regions that open very early in all cells. So that would be one place that I would look at. Okay, so that's another interesting thing that we can refer to in our review. It relates to early reprogramming. It probably have consequence for rejuvenation. We can say we don't know which group, but we can say we know overall what's happening to different groups. And one more thing from this paper, the key finding is on, they find new insight on DNA methylation. So before this work, almost everyone disregard DNA methylation as something that's happening after the transcriptomic wave, after chromatin remodeling and transcriptomic wave, and as a kind of memory marker. But looking deeper based on these enriched cells, based on these POIT cells, they still confirm that most of the cells, so we have all the cells here, and this is the unbiased clustering based on how their DNA methylation changes. And most of the cells are like that. Most of the clusters, they are methylated. They are the unmethylated regions. They stay unmethylated, and all the way at iPSC, they become accessible. They become unmethylated. But two clusters are different. They are methylated in somatic cells in fibroblasts, but one of them around day 6, especially in the efficient, become less methylated, and this is a region that needs to be completely unmethylated in iPSC, and another one that needs to be unmethylated in iPSC, it starts becoming demethylated early on. So last time we talked about this, Vittorio, we thought our regions mostly overlap with pattern-wise, mostly look like a cluster 10, but I looked at cluster 9, and our regions also are in cluster 9. And based on a quick analysis of our region of interest and how their DNA methylation changes, it seems day 6 efficient is where we see the more changes, and this pattern corresponds more closely to cluster 9. So that's one thing that I'm going to look deeper, and that's one piece of the paper that directly relates to our empirical study of epigenome. So in that sense, this was also an interesting paper. And interestingly, the other group of people that are studying epigenetic reprogramming through deterministic reprogramming solution. If you remember, we said there are these IPS poised cells and this deterministic approach. We can also study the epigenome, and based on that data, we also know that DNA methylation started to change early on. So based on all these, I think we can safely conclude that DNA methylation also changes early. This paper conclusion was the methylation and DNA and then demethylation are uncoupled, and the methylation happens late. The methylation happens late, but the demethylation could start from early. Yeah. And this was also another new finding of the paper. That
sorry, Matti, deterministic reprogramming. So if I see the first histogram, the second one is just a time course from math to iPSCs, but the cells were not sorted. That's the difference.
So in deterministic programming, it's a different approach. They change the protocol. They add more chromatin remodelers to synchronize the reprogramming events.
Okay.
So
it's like all of them become efficient. Basically.
Yeah, to
it's
find
like
more
looking
efficient.
all the efficient.
Exactly. So
that's
it's a
why
more
the phenotype is way stronger than the efficient versus inefficient.
The idea is to remove a stochasticity based on proposing more efficient approaches for reprogramming. And I think both of them are great tools to study. They are like microscopes for looking at reprogramming. And I think these kind of platforms could help us to understand the same way it's helping them to understand early reprogramming for pluripotency. It could also help us to understand the rejuvenation effect of reprogramming. And probably we either need to at some point follow some of these protocols or collaborate with them. So this group is very strong. That's the group that I'm presenting their paper today. It's from now they are collaborating. They are also co-authoring with Broad, which is an institute of MIT Harvard. And I'm considering at some point to collaborate or work with them. So I think it's a good opportunity to use these platforms. And in summary, from this paper, they look at somatic locus and pluripotency locus, and from embryonic embryonic fibroblasts all the way to iPSC, focusing on the earliest stages, day 3 and day 6. And they divide the cells into efficient and inefficient based on facts. And they find out that within the efficient cells, chromatin accessibility changes and in somatic cells, and the regions that are accessible, they immediately become closed only in efficient cells. And for DNA methylation, not much change in day 3, but in day 6, they see that especially in pluripotency regions, they become accessible early on, and they also lose methylation, the demethylation happens in efficient cells, definitely by day 6, but even in day 3, they suggest that even in inefficient cells, there's some loss of DNA methylation early on. So one of the other findings was that the demethylation has a role in early reprogramming. And that's all we have. And I also want to specifically thank Andy for all the help with this work, and Sasha and everyone else who helped with the work. And if you have any remaining questions, we can discuss that.
Man, can you walk like back? Sorry. But I think also here it's crucial with Vittorio's point, because if you see the inefficient one, it's okay. If you want to do a rejuvenation, I'm just thinking loud here, you don't want the somatic locus to be closed. So you would like to have something which is like close to the inefficient. But in order to have a rejuvenation, that region needs to be like probably, I don't know, clean in order to allow a better, just very basic, oversimplifying system expression of those genes. So I think the question, I think there are two models in my mind. Now, the first one is, okay, in order to achieve a better chromatin landscape for young cells, I need to undergo into closing it and opening them again, which is like the hypothesis where we need a partial differentiation in order to go all the way back. And this is demonstrated because if you do IPSCs and you redifferentiating back, all the aging markers are erased. So this is known. On the other side, in the inefficient way, it's basically a condition where you are not affecting the somatic loci, but you still might do some cleaning. So I think a key point that we should address in order to understand if the inefficient population are rejuvenating is that if it's possible to achieve a better chromatin landscape without undergoing into this partial differentiation. And I mean, looking at, again, as Vittorio was saying, David's paper, Belmonte paper, it might be the case, right? Because if you're working in a post-mitotic cells, I don't think that you are like erasing the somatic loci, changing gene expression, and then pushing the cell back. So for the in vivo reprogramming, probably it might be reasonable to think that you can achieve rejuvenation without undergoing to this closing and opening again. I don't know if it makes sense, because it's not clear in my mind as well.
Yeah, so here I would say day 3 inefficient are not in a big sense different than fibroblasts. So I would say these are the cells that we could not influence much, but these could be the cells that some somatic TFs have been displaced, but we are going against the current, and these cells strongly want to get back to their original
which
network.
Makes sense. So, which makes sense with the precursor redifferentiating back. Yeah,
so I don't think we are talking about cell identity here. If you even 10% are a good percent of the cells, if you take their TF out of their place, I don't think they lose their identity. They just reproduce more TFs and they just find the same place because there are thousands of epigenetic marks. There are many histone marks that remember what is this cells. And you remember even in the fibroblast, we lost half of the DNA methylation, but still they are inaccessible regions because of probably other histone marks. So I would say there are a lot of redundancy in epigenetic memories. And I don't think removing a few transcription factors or even part of them would lose cell identity based on
cell memory. I would
lines.
say probably cell memory.
Yeah, identity is probably a loose term, so we still don't know what is cell identity. But considering that a fibroblast cell line is still fibroblast after going through all that and losing that much methylation, I think, and so personally, I feel it's good to go to replace somatic TF and to at least to some extent go through the path of reprogramming to see the effect. And the good thing is we know we are going to see some effect here, but here we might not see any effect. So I think if we are designing experiment, it's safer to do some reprogramming effect, and at least for the early few days to look at the path toward reprogramming. I think it's more promising. And there might be similar effects to a lower extent in the other path, but we have a better resolution at least.
Another thing that I was actually thinking about, and this is something that I think we wanted to initially include in the review, and then we ended up not because it seemed a little less relevant. But the studies that show that you can actually directly differentiate or directly reprogram cells from fibroblasts to neurons, or so completely different cell types without undergoing the stem cell state, that actually maintains the age signature, right? So it's basically going through cell identity change without resetting the epigenome.
Yeah.
And I don't
this was in the introduction, but that Ryan said I wouldn't put it, but I strongly disagree, because I think it's a key point here, because it's true that it's dangerous talking about the differentiation, because we want to point out that partial reprogramming, but I think that based also on this evidence, I think it's essential to make people understand that probably this partial state, with Maddie, we talked a lot about this 2 days ago. I think it's important to underline the fact that probably this partial differentiation that maintain the memory might be probably a necessary condition to achieve rejuvenation. And the evidence is that exactly what you said now. If we trans, no change. If we de-differentiate to iPSC, so the extreme other condition, and we re-differentiating, all the aging markers are reset. So it's still in the introduction.
Right. Yeah, because I think there is probably a hierarchy of the networks that are being activated with the pluripotency program, and it might not necessarily push the cells completely to a pluripotent state, but there is a base layer that you need to activate in order to rejuvenate the epigenome. And that, I think, is sort of decoupled from cell identity programs. And just because we can change the cell identity without resetting the epigenome with the age signature. Yeah, I think it's an important point, but I'm not really sure how to kind of incorporate it without kind of going off the deep end. So that's something that we'll try to kind of address throughout the document, I think.
All right, any other questions about the paper before I stop recording? To have this as an independent piece on reprogramming events.
I personally think that we should comment on, maybe in a short format, we should comment about the fact that nuclear reprogramming per se is not a prerequisite of rejuvenation, but it can happen only with a specific set of factors. Now, again, the question remains open. Is it because the Yamanaka factors, because of the deep differentiation, are also doing that? Possibly. Or is it because they're binding, for whatever evolutionary reason, they're binding to very specific spots of the genome that happen to be involved both in the de-differentiation, but also in the rejuvenation, we still don't know.
I agree.
Because if you think also about it, I mean, just playing devil's advocate here. I mean, if you were to overexpress KLF4 and CMYC alone, those are expressed in a variety of different somatic cell types, right? But their expression doesn't necessarily mean that the cells are younger, right? They age, and they age still being KLF4 positive and CMYC positive. It is the unique combination of the factors that is doing something. So it's not the factor alone, or maybe it's not the factor alone that explains, but it's the unique combination of those factors. And you can probably argue that really OCT4 and SOX2 are the main drivers.
Yeah. Yeah, they're the only pioneer ones, right? So they're the only one able to displace nucleosomes.
Yeah.
And Madi, right, there are also iPSCs done with OS. You remember you told me that there was like an article where they were able to do iPSCs just using ONS.
Especially if they have other factors intrinsically internally expressed. So some cells, they have other factors that are expressed internally. You give them the rest of them.
Even SOX2, SOX2 in combination with other factors can be used to generate, you know, I guess if I remember correctly, neural stem cells or something like that. Now, does that mean maybe the neural stem cells are younger because they're stem cells? So nobody has ever looked at that, for example.
Yeah, the only factors with confirmed chromatin remodeling activity is OCT4 and SOX2. The rest, we know they are helping in some way.
I think it's also kind of difficult to argue, though, that pioneer factor activity is like the main driver of the rejuvenation effects, because with direct reprogramming of fibroblasts into neurons, one of the factors that you need is ASCL1. And ASCL1 is a neural factor, but it's also a pioneer factor, yeah. Right? So it's not necessarily the pioneer factor activity itself that's driving rejuvenation. That's more in general, if you're going to reset the epigenome in whatever, I guess, context, that's more important. But I think it's unclear why some pioneer factors would reset the epigenome age versus the cell identity. I think that's the thing that's like pretty unclear.
Yeah. Also the binding side, right, is important, because I mean also right. In muscle transdifferentiation with MyoD and BAF60 produce muscle, because MyoD gives the specificity in that case. So when you told this, I was thinking about MyoD and BAF. Now BAF is a chromatin remodeling factor. When fused with MyoD, which is probably the most specific muscle transcription factor, because it's expressed only and in all skeletal muscle cells, you transdifferentiate, so you give specificity in that case. So I think probably an answer, speculation, might be that the binding motive of OCT4 and SOX2, since these factors are needed in a stage where pluripotency, which means everything open, everything poised, is important, might in some way drive the epigenome in a more broad way, compared with a very specific pioneer factor, which is also FOXA2 is a pioneer. And if you overexpress FOXA2, you push the cells in the endoderm and pharyngeal endoderm stage. So I think a key answer might be the binding motive that gives the specificity to the pioneer function, which is pretty universal. Probably. I don't know if it makes sense.
Cool.
Okay, let me first stop the recording, of course.